presentation. I speak Russian, Hebrew, and English, but not a single word in French, so I'm sure that your jokes were very funny and I can relate that. Before I start, I must confess polygamy. And unfortunately, it's not as exciting probably as being married to several women. I mean academic polygamy. And that is, I am truly and fully committed to 2 universities. And one of them is UCLA in Los Angeles, the other one is Bar-Ilan in Tel Aviv. Unfortunately, they are separated by a couple of continents and a 15-hour direct flight. So naturally, I do somewhat different research activities in each of those 2 universities. And what I'm going to talk today is mostly related to my work at UCLA. And for those of you who will join us later or just are interested, I can tell you more about my research at Bar-Ilan, which is not less exciting, but it's somewhat different. Okay, so let me start by a brief outline of the presentation. And I did not know the heterogeneity of the audience, so I have some things are a little basic, and I will not go in depth. I realize that mostly are graduate students and faculty, so I will talk briefly about things that have to do with sort of the diagnostic criteria and things like that about schizophrenia. I will try to focus more on my studies. I will talk about cognition in general, how we ended up talking about cognition in schizophrenia. This is not a trivial issue. And most of this lecture I will spend about the research that is conducted at our lab. With a few remaining minutes, I will talk about what I plan to do in the future. Now, please interrupt me during the presentation. I don't have to finish everything in one setting. I can stop and answer your questions. I like when this is more interactive rather than just boring you to death with an hour and a half of spitting out everything I have. Okay, so let me start by making a fairly basic distinction when the DSM just started in sort of in the context of psychodynamic approach. There was this basic distinction between neurotic disorders, which one of my favorite sitcoms probably demonstrates it best. George Costanza is your typical classical neurotic where he can hold a job, he can think about things reasonably, but he's just a very anxious individual. Versus the psychotic disorders, and as you might have guessed, this is our guy there, Kramer, who is completely detached and he sort of lives in his own world. This was the basic distinction between neurotic and psychotic disorders. And then it became more complicated. Started first by Emil Kraepelin, who worked along with Alzheimer, and hence this term of dementia precox to distinguish it from senile dementia. Senile dementia was considered the dementia that starts later in life, 65 and up, and dementia precox was the early dementia. So Emil Kraepelin actually thought of schizophrenia as a cognitive disorder in a way. And then Eugene Bleuler sort of confused the conceptualization of schizophrenia because the definition, the literal definition of schizophrenia is split soul or split mind. And so whenever I ask my undergraduate student during their first psychopathology class, what is schizophrenia, they tell me it's multiple personality. So there is something split, but this is not what Eugene Bleuler had in mind. He had in mind actually splitting of various functions within sort of the different affect, different cognitions, so internal splitting of some sort. But this made us refocus our conceptualization from cognitive aspects of schizophrenia to more of a psychotic conceptualization of schizophrenia. In terms of epidemiology, fairly young in age, 16 to 25, it happens in women a little bit later than in men. If you pass your 30s or 35, the risk drops down tremendously. And it affects about 1% of the population wherever we go. In DSM-5, this remained more or less the same as it has been before. The key change was that now you have to have at least one symptom of these three. Okay, DSM-4, you could have any two constellation of symptoms. DSM-5, you have to have at least one of the three, and then you can have either the first two and one of the 4 and 5. Okay, and the other major thing they did in DSM-5, they dropped those fascinating categories of subtypes. Paranoid schizophrenia, disorganized type, catatonic type, no longer exists. Okay. And of course, across all diagnostic categories, there has to be some impact on their lives. The way we thought about schizophrenia historically since Eugene Buehler is in terms of positive and negative symptoms. Positive symptoms doesn't mean it's something good, it's just that there's something added to our experience. Okay, disorganization, hallucination, delusions, all these cool symptoms. Negative symptoms is a lack of something. Okay, so something is lacking, flat affect, reduced affect, inability to experience pleasure, things like that. And what happened over the last 30 to 40 years is there is an increase in interest in the neuropsychology of schizophrenia. Now, away from the symptoms and more on the cognition. As you can see, there is an exponential increase in interest over the last 30 to 40 years. Now, how can we explain this shift in conceptualization? So, although we might like to think about science kind of in Kuhnian terms, you know, there's more and more evidence until there's enough evidence, and then there's like a breakdown of a major theory and so forth. It doesn't necessarily work that way. It's sometimes science works in a circular way. We started from Emil Kraepelin, thought about cognition, then went with Buehler, so thought about symptoms, and then returned back to cognition. Now there are three basic aspects that led to this reconceptualization of schizophrenia. One in terms of the core feature of the illness. Cognitive symptoms present before the onset of clinical symptoms. Before the outbreak of schizophrenia, we already see some deficits in cognitive functions and at-risk individuals, twin studies, relatives, and all that kind of stuff. We see them in unaffected first-degree relatives. So even if we have one family member with schizophrenia, the other ones can go about their life never having any schizophrenia symptoms, yet they still show some cognitive deficits. They're relatively stable across the clinical state and lifespan. So even when there is effective treatment with neuroleptics, they still continue to show cognitive deficits. Low cross-sectional correlation with psychotic symptoms, you'd think, okay, so maybe it's part of psychosis. Well, it's not. Cognitive symptoms seems to be something different from psychosis. There is discrepancy between clinical and cognitive effects of antipsychotic meds, so the medication can affect the psychotic symptoms but do nothing to cognition. There is also a relevance of cognition to the genetics of schizophrenia. As Steve Hyman, cited in Biological Psychiatry, said, while the DSM-4 is a useful tool for communication, it would be foolhardy to think that its criteria select anything that maps onto the genome. Okay, now there is actually NIH initiatives to move away from those diagnostic criteria and think more about broader spectrum of symptoms. Some cognitive deficits reflect predisposition to schizophrenia rather than presence of clinical symptoms. And they can serve as endophenotype, sort of an intermediate step between genes and manifestation of symptoms. And these linkages provide a rational approach to identify new neurochemical agents. Okay, so we know about memory, we know where in the brain certain mechanisms of memory are operating and how they're operating. And so we can zero in on these specific areas and look there for different genetic linkages. What about functional outcome? Cognitive deficits are reliable correlates and predictors of functional outcome. The more somebody is impaired cognitively, obviously they have more disability. Associations are medium for specific constructs like memory or retention, but when you take summary scores, like for example, the Wechsler IQ general score, it's not necessarily a good example because not necessarily the IQ, but other neuropsychological measures are more relevant. But that's what I'm talking about. If you take just a vocabulary score and try to predict outcome, it would be a medium association. If you take the overall IQ score, the association will be much larger. Relationships are stronger than between psychotic symptoms and functional outcomes. Okay, so it seems that the impairment resulting from deficits in cognition are much greater than from psychotic symptoms. So somebody might believe, for example, I don't know, that the FBI is out there to get them, and he might have this delusion, and well, you know, the FBI might actually be after them. But if he is in fact schizophrenic and has this delusion, he could still somehow function in society. You know, he can sort of walk around the block, try to remove whoever is following them, and get to work and do the things they're supposed to do, go back home, live within this delusion. But once he gets memory problems, executive deficits, all this kind of stuff, he no longer is able to function effectively in society. And they are linked to success in psychiatric rehabilitation. Okay, so very good predictors of psychiatric rehabilitation. All this result, you don't have to read all this stuff. This is sort of for me to pry me into the correct, into the right direction. All this constellation of research led to a very interesting development for us as neuroscientists and neuropsychologists in this area of schizophrenia. What it did is actually bring the representative from the key bodies in the US. It's the FDA, the federal government representative, the pharmaceutical representative from the pharmaceutical industry, and representative from the academia. And all these representatives sat in one room and decided, okay, now it's time to start and think about the cognitive deficits in schizophrenia. And the fact that they are relevant, we now need to start making medications that would improve cognitive deficit in schizophrenia. And for this, what needs to be done is to create some measure, some reliable measure of cognition that we will base our pharmacological agents to improve these functions. And the resulting is this matrix consensus battery, and this is actually a constellation of various available and several novel tasks that came together, and now this is a neuropsychological battery that is used for pharmacological agents to see if it does anything to schizophrenia patients, any benefit to schizophrenia patients. Now, I'm going to very briefly give you some examples. For those of you who are not familiar with clinical neuropsychology, I will very briefly show you the kind of things that we use. Measures of attention, information processing speed, working memory, verbal and nonverbal, learning and memory, planning and reasoning, and social cognition. So some typical examples, this is the processing speed. I'm sure you know the trail making test. They need to connect the circles in order as fast as they can, and we measure the number of errors and the time it takes to complete this. This is an example of immediate working memory. This is of the ways, letter number sequencing test. We read the stimulus, and then they need to rearrange with the numbers from 1 and up and the letters in alphabetical order. So sort of needing to keep online the information online. And this is the counterpart for the working memory. This is a visual working memory. Over here, we basically press these in a certain order, and they need to press them kind of like a game, the Simon Says game. The verbal learning and memory tests, there's a whole bunch of the list learning tests, many languages and many forms, and basically you read the list several times. They have to repeat it several times, so you can generate the learning curve, you can examine the recognition, the recall, and all these kind of stuff. Visual learning and memory, again, the counterpart of the verbal learning and memory, showing some pictures. and then you cover, and they need to reproduce it from memory, then they need to recognize it from several other stimuli. The card sorting test, you have several stimuli cards. Now it's done on a computer, but how many of you knew that actually for clinical purposes you cannot do this on a computer? Because if you look at the output of the Wisconsin on a computer, it says for research purpose only. So it hasn't been standardized apparently, even though Heaton already has a whole bunch of newer versions. Still, you're supposed to, for clinical use the cards. I don't know that many people who actually do that. But you basically need to match it according to color, shape, or number, and you test things like flexibility, shifting percent, and stuff like that. And the final one is test of planning. You have here a stimulus form, and you need to arrange these beads in as few as trials as you can to place them according to the stimulus. So these are the kind of tests that we give to examine the cognitive functions in schizophrenics. And in our lab, as you can see, there is a fairly large spectrum of different research questions that we ask, from normal neurosystems, social cognition, and innovative psychosocial interventions, phenotypes for genetics, interventions for cognitive deficits. I'm not involved in all of them. Okay? This is our entire lab at UCLA is involved in all these things. I am going to focus actually on the things that I do, with some of the things that I'm more or less involved, but coming from the general people in our lab. So I'm going to talk actually about the three kinds of studies. Studies in basic cognition, which is mostly I will focus on visual processes. This is my own interest. I will talk about social cognition. I will talk about dysfunctional cognitions. And then the last few minutes I will spend about cognitive enhancement that has become my interest more recently. And this is the grant for which I received the award from NARCET. And I will tell you a little bit about that. No results yet. We get the money yet. Okay. So visual perception in schizophrenia. The way I've been studying visual perception in schizophrenia through the technique known as masking, and there are many, many paradigms that use visual masking. Basically what you do is you show a stimulus, and a very brief, a few milliseconds later, you mask it with something that covers either partially or entirely the stimulus. And in this particular paradigm, you just ask the patients to tell you if the hole is on top or on the side and so forth. Okay? And when there is basically a very short time between the stimulus and the mask, it interferes with their perception. Okay? And when we do something like this, we find very, very consistent. By the way, there's almost no practice effect. So it's a really nice paradigm because you can repeat it several times. You can track their progress on medication or anything like that. Okay, so it's a really nice paradigm. But we see very consistently, we repeated it several times. As you can see here, patients do worse than controls. I will not bore you with technical details about that, but just to understand what is going on here is that we have stimulus onset asynchronous, which is the time interval between the onset of the stimulus and the onset of the mask. Okay, you might have heard about ISI, interstimulus interval, which is a different measure, a similar measure between the ending of the stimulus and the beginning of the response, but it's pretty much the same. And the negative numbers here means that the mask comes before the stimulus. Okay, the positive numbers is that the mask comes after the stimulus. But both ways, as you can see, patients do worse. Another interesting thing to note here is that the maximal suppression, this is where both patients and controls perform the worst. And this is not here. This is not when they appear simultaneously. This is actually about 40 milliseconds separation. And this means that the maximum competition between processing of the stimulus and processing of the mask happens at this point. Okay. So being a clinical neuropsychologist, of course, the first thing that comes to mind is a what. Okay, this is basic kind of science, and you know, people did it before me and did it not in schizophrenia and hormones and fine, all this kind of stuff. So why is this interesting? Well, it is kind of interesting because when you look at what these deficits predict, and I'll talk more about the study design later, but right now I'm just going to give you sort of a prelude of what I'm going to do later. This is interesting because we actually were surprised by this finding. Early visual deficits in schizophrenia predict not a large proportion, so this is like a regression coefficient. If you square it, you'll get about 12% variance explained. It's not much, but it's kind of amazing that early visual perception deficits predicts functional outcome, how they do in work, how they do in their daily lives, and how they do in their social relations. So we didn't expect to find any significant relationship here, but we did. So we ended up trying to investigate what might be the neural mechanisms responsible for these visual deficits. And as you know, when you look at these textbooks from undergraduates or even graduate textbooks, the largest portion or the largest proportion is devoted to visual processing, because we just know quite a bit about that. We've been studying for a long time. There's been increased interest. This is on our primary senses, so we're devoting quite a bit of efforts to understand vision. And we know that from the retina, information travels to the thalamic nuclei, the LGN, and ends up at the primary visual cortex, and from there it sort of feeds back and starts to develop more complex visual information processing aspects. Okay? Now, this separation is, these pathways are being fed by two types of cells. The magnocellular cells, which are large, fast-acting, have a very high temporal resolution, they act very fast, and they sort of do the first gross dirty work. They help us separate objects from background. So you might notice here the dog sniffing the ground. So just sort of very basic initial capturing of the object. And then we get into the more higher resolution parvocellular cells, which yield more of a detailed processing. Okay, those fire slowly and help us process information in a slow and more precise manner. And it turns out that you can bias the visual system by displaying the stimuli on a red background. We learned this from basic research, we learned this in animal research, and in normals, that when you show something on a red background, it actually inhibits the magnocellular cells primarily. Okay? So we sort of repeated the same experiment, but now what we did is we displayed those visual stimuli en masse on the red background with the assumption that something different will happen when we do something differentially to the magnocellular versus parvocellular cells. Indeed, we found something quite interesting. We used, by the way, as a control condition, we used green background because the green doesn't have any red in it. Okay, it could have been gray or anything else, but we just used the opposite. Red is green. So if you notice here, an interesting thing is happening. When we look at controls, when they view the masking stimuli on the red background, they actually perform better. Okay, they perform better than when they do it on the green or control background. The exact opposite is true for patients. When they see stimuli on the red background, they do worse. Okay. Give you just 1 second to process this because it's yeah.
Was the difference between the two groups on the green background, was it significant?
Yes.
So So the control were still better.
Right. And I mean, we have a theory of what happens. Just wonder if you want to like try to come up why this is true, given what I just told you about the magma and parvo cells and how masking works.
Similar to autism?
Why did that be?
Look at local versus global details.
Kind of. Well, I mean, it's possible. This is not what we thought, but it's possible. What does masking do to the stimuli? Masking suppresses the processing of the stimulus. And now, masking is something very brief that primarily activates the magma cells. What do we do with red background? We inhibit masking. We primarily inhibit masking. And that's why you sort of see a reversal here. Okay, so less masking, better performance. With patients, something odd happens, fairly consistently, by the way. This has been replicated in several other studies. They do worse on red background when you mess up with their magma cells. They do even worse. And you know, your guess is as good as mine. We try now with fMRI, but those results are not available yet. It is possible that their magma cells is dysfunctional in some way. The hypothesis here that they're basically hyperactive, right? They're hyperactive, that's why suppression of magnocellular cells reduces their performance. But we don't know exactly what happens. Another interesting thing, just to mention that this deficit with a red background or deficits in magma cells predicts various interesting things like negative symptoms, estimated IQ, and their performance on a Stroop. So these deficits, again, relate to certain symptoms and certain cognitive abilities. Okay, so this is for the basic vision research. To summarize it, we find that individuals with schizophrenia have visual perceptual deficits, and these deficits detrimentally affect their daily function. Okay? Let me, unless you want to ask me for any clarification, just in comments, I'll move to the social cognitives. Okay. So, social cognitive perception deficits is another aspect that exists in schizophrenia. In social cognition is defined as the ability to construct mental representation about others, oneself, and relations between others and oneself. And it is thought to facilitate skillful social interactions and is considered a potential determinant of social dysfunction among schizophrenia. All this research, by the way, about social cognition came from autism research, which predated it by quite a bit, and you reminded me of this because there was a large body of research demonstrating social cognitive deficits in autism, and we sort of borrowed many of these tasks and used it in schizophrenia. Now, social cognition is a complex, abstract, latent variable. There's a lot of stuff going on. Think about it. This is social interaction. You live in a complex environment. There are many things that are happening both internally and externally. So it is a fairly complicated, you can, I guess, sort of analogously compare it to an IQ. IQ consists of many different components and concepts. Same goes for social cognition. It depends on social knowledge, on social perception, in context, on relationships, emotion processing, you need to manage your emotions, and affect, you need to monitor and adjust your affect. Theory of mind, it is more and more becomes known as mentalizing, and attributional styles. And I don't know of any other components, but some people might come up with some other components that might be added to this conceptualization. So in order to do work in this, I had to learn an interesting methodological technique, and I've been using it since. It's called structural equation modeling. I'm sure that all of you heard about structural equation modeling. I published my first paper on SEM about a decade ago, and it took me over a year to publish it. The editor just couldn't find a reviewer to review because nobody knew what structural equation modeling was. Luckily, everyone knows it, and I like this methodology. It sort of helps me think in complex terms about research. And what you do here is basically, it's a family of statistical techniques. There are many different types of software that can be used to do this. But they all share one thing. It consists of two steps. Validating the measurement model, which is analogous to confirmatory factor analysis, and then fitting the structural model to path analysis with latent variables. So conceptually, it's sort of a combination of factor analysis and multiple regression. But now you have a bunch of different equations estimated at the same time. Okay? It's intuitively, actually, it's quite simple. Okay? It's complicated to think about it statistically, but intuitively, it's quite simple. So what you do here is you have two steps. This is the measurement model. So this is the same as factor analysis. You have a latent variable, and you have a bunch of measures. Think of it like an IQ. So this is your overall IQ, and this, for example, vocabulary, information, and similarity. Okay? So several measures of a construct, and this is something else. The interesting part here, of course, is the second part. That's the structural model, which you can directly test whether one latent variable is related to another latent variable. And now you can ask questions about, not only about, for example, does vocabulary score predict, I don't know, negative symptom score? You can ask questions like, does IQ relate to functioning in daily life? So you can actually speak in abstract terms with it. And it is powerful statistically because it actually maximizes true variance. Okay, maximizes true variance. Just think about it, because the variance that is loaded onto here is all the common variance shared in here. Okay, so it maximizes true variance, so it is more powerful than just using multiple regression and having several variables estimate an independent variable. Okay. So started doing this. This is back to my original slide when we found that early visual perception affects functional outcome with fairly small variance explained. And the question is, can we improve this model? Again, thinking as a clinical neuropsychologist, it's very difficult to do something here. Right? Early visual perception, you sort of perceive the environment on a very basic information processing model. And you cannot do that much intervention there. But what if you were to add social perception, okay, in there? And what we find here, that social perception significantly mediates this relationship, or that, in other words, what actually explains the relationship between this and this is this. And this is a full mediation. It could be partial mediation. The fact that this dropped to almost zero means that this is a full mediation model. That the whole variance here is explained by this. Okay? And this was a measure. We showed patients various vignettes, and we asked them to tell us what the characters were feeling. Okay? So basically, they don't process basic visual information very well, which in turn impacts their social skills, their ability to extract emotional content from stimuli. And this in turn leads to their functional deficits. This model is much better, much stronger, and it gives us a way to do something about it. We can teach patients to do these things, right? We can show them, for instance, one problem they have is that they're unable to recognize different affects. When somebody is happy, somebody is sad, they're not very good at this. We can show them, and there are actually right now computerized programs, and there are interventions, one-on-one intervention, that they show patients, okay, now this phase is actually, the person is happy. We can do something in terms of rehabilitation there. Okay. Again, given, yes?
Excuse me, do the arrows represent the causal relationships between one?
This is an excellent question. And causal is not, yes. The simple answer to your question is yes, but this is not what impacts causality onto the model. It's the experimental design. Remember, to your undergraduate statistic, it's the experimental design that allows you to infer causality. So although these are directional arrows and we would like to think about them as causal models, it is an unfortunate incident that people call those causal models, because there is nothing inherently causal about them. Only if we are to do an experiment, well controlled, with random random distribution of control and all that experimental condition, only then we can infer causality. But this is our hypothesis. That's why in structural equation modeling is critical to come up with a good theory. Okay, so you should be able to, before you test this model, you should be able to theorize about it. Why would this be like that? Why would you expect this to be like that? Just like I did before. Okay, just like I did before, I told you that probably basic visual information, impairments in basic visual processing might influence the way they extract emotional information from the environment, which in turn impacts their functional outcome. Okay, but you are correct. Another thing that I would like to mention about these arrows, that this is sort of like partial correlation coefficient or incremental coefficient. They explain the unique variance for each of these. So, in other words, it doesn't mean that this is not important. If I take this out, there is a significant relationship. But when I put this in, the unique contribution of this to this over and above social perception is zero. Okay. Okay. There is I'm going to show you a famous experiment which many of you may be familiar with. And the reason I'm doing this then is that my next ideas came from this experiment.
The monkey business illusion.
If you know this, don't say anything.
Count how many times the players wearing white pass the ball. The correct answer is 16 passes. Did you spot the gorilla?
How many?
For people who haven't seen or heard about a video like this before, about half missed the gorilla. If you knew about the gorilla, you probably saw it. But did you notice the curtain changing color or the player on the black team leaving the game? Let's rewind and watch it again. Here comes the gorilla, and there goes a player, and the curtain is changing from red to gold. When you're looking for a gorilla, you often miss other unexpected effects. And that's the monkey business illusion. Learn more about this.
Okay. So this was sort of the idea for the next study came based on this and similar study. The very fact that top-down processes, your expectations, the way you expect to conceptualize and process the environment influences how you actually see it, made me think of the following study. And we actually know, again, from animal and basic vision studies, that there are, in fact, so-called reentrant processes that probably go as low as the thalamus. So there are feedback mechanisms for our visual system that set up your expectations and influence how you see the environment, which is going down to the thalamus, is pretty low. So the way you see the world is influenced quite a bit by your expectation. So what we try to do here is try to disentangle this deficit of social cognitive processing in schizophrenia in terms of whether it's a bottom-up information processing deficit or rather top-down information processing deficit. This is one condition of the experiment. So what we did here is we placed a TMS coil. This is transcranial magnetic stimulation, a coil with an electrical field that generates magnetic pulses. And when you place this on V1 and give a single pulse, it interferes, it creates temporary lesion. Schizophrenias are okay with that. You know, you think with all the delusions, try to put them in a PET scan. You tell them you're gonna right now inject in their vein something chemical. But this one they're okay with. So we placed this, and we actually simulated a masking study, but now with TMS. The advantage is that right now you're creating exact lesions at V1. And by manipulating the timing of the TMS administration, you can actually separate the feed forward, or forward masking would be regarded as feed forward. You first give a TMS pulse, then you show a stimulus. A feedback mechanism happens later in the process. So when you show them the stimulus and several milliseconds later give them the TMS pulse, it would be a feedback interruption. Okay? And of course, we haven't forgotten, we haven't forgotten the magno versus parvo cells. So we had another condition there, is that we manipulated the spatial frequency of the stimuli. So basically, those are again basic vision research kind of paradigms. When they show stimuli like this one with very high spatial frequency, which primarily affects the parvo cells, and when they show stimuli with low spatial frequency, it mostly affects the magno cells. So we did the exact same thing with faces. We showed them faces and did a systematic manipulation in MATLAB where it allows you to parametrically set up the spatial frequency for the stimuli. Okay? So this would be biasing towards the magno system, the low spatial frequency, and this would be biasing where you see mostly the details, but you don't see much of the general outline. This would be biasing for the parvo cells, and of course this is a normal control condition. Well, a long story short, we got money for it from an NIH grant and some money from the NARCET, got an investigator award. Those of you who are graduate students, I keep telling my graduate students when they're very concerned about positive findings. I tell them, you're not responsible to demonstrate positive findings. You're only responsible to do a good study. Okay? This was a good study, positive findings. So now it's easier for me to tell them. So I spent quite a bit of taxpayer money and didn't demonstrate positive finding, but I keep convincing myself this is still important. It was informative. You know, I shouldn't reduce too much of the story because the finding was important, and you'll see why. By the way, it's very labor intensive to do precision TMS. To find the exact spot on the V1 and stimulate it and suppress vision precisely, it's very, very labor intensive. We actually had to develop a new methodology for this. Usually what they do is they place, we have Bruno Brettmeyer is a world-renowned vision researcher, basic vision researcher. So we collaborated with him, and he told us how to do this technique. What they basically did is place the TMS on V1 and ask the participants whether they see phosphines. Phosphines are sort of brief flashes of light. Okay? If they see the phosphines, it meant they got the V1. We tried it, didn't work. We tried it again, it didn't work. And then I called Bruno, what's up? It doesn't work. And after sort of an hour conversation, he told me, well, you know, it was our lab team who did the experiments. Maybe that's the difference. You have a bunch of schizophrenics. We are, you know, brilliant lab researchers. Maybe there is some difference there. Maybe these people see the phosphines much more readily than your, you know, odd subjects there. So we had to develop our own mechanism to find the right spot. And we called it the hotspot procedure. We fitted them with a swim cap and put a grid around the visual area, around the visual cortex, and started stimulating them along this grid, a little bit up, a centimeter down, until we found the hotspot by giving them a behavioral procedure. We showed them a letter triagram. And when they stopped recognizing the letters effectively, we decided, okay, we're at the hotspot. Okay? So we did do something useful for future research. This is the negative findings. As you can see, well, you know, patients perform worse. This is not mind-boggling finding. Patients perform worse, but as you can see, we expected an interaction effect. We expected an interaction by group effect. We wanted to see something different happening with the top-down versus bottom-up, or with the magna versus parvo. We did not find this. Okay? Probably the visual masking, this competition between two stimuli, does something different to patients than lesions in the cortex. Their brains are not fundamentally different enough from controls to detect this difference.
Why no phosphine?
Say again?
Why no phosphines in schizophrenia?
Why no?
Why did you get this? Why did you replicate the results for finding V1 with the schizophrenics that they found with brilliant graduate students?
Well, we did replicate, but they were just not different than controls. We replicated with the U procedure.
No, but why couldn't you replicate it? Do you have any hypotheses why you couldn't do it?
I don't know. After that, I spoke with different researchers, and apparently it is a very labor and difficult procedure to do precision stimulation with TMS.
And schizophrenia.
In general, yeah. I mean,
Did you off-target the schizophrenia?
well, I'm not sure. You know, again, you sort of start a study, you run it, you become consistent, then you reach the end, and that's what you have.
Because there may be something basically different. Could be basic. There may be something basic.
Well, you know, they do show the exact same. These are the controls and the patients. They show pretty much the same processing of information here. So I'm not sure. Yeah.
Did you have any schizophrenic reporting phosphine in any
Yeah,
kind of situation?
Yeah,
okay.
It was just not consistent.
Okay.
You know, there is another thing that I should mention is that there is only as high you can go with the TMS machine. You're sending a pulse, and when you reach a certain level, it becomes painful. So you cannot do that very, you know, it can cause seizures, but you know, the we were okay with that because it actually also used as treatment for seizures. So it's okay, it reduces the threshold for seizures, but single pulse is okay. It's usually the RTMS, repeated TMS, when you do several in a second, very rapid TMS, then there is a higher risk. But single pulse TMS is still, you don't want to, also actually when you go a little bit too low on the cortex, on the brain there, go towards the spine, you start getting sort of jerky movements. So it's uncomfortable, and they don't want to do it, and I don't blame them. So yeah, they at a level where we had the threshold, it was inconsistent. Some saw phosphines and others didn't. But I think the behavioral measure, this is actually a good thing. Now you can objectively test whether they whether we're on target or not. Yes?
I want to get on this thing, but the phosphine is kind of an activation of the brain activity, and for your hotspot, it seems that you localize the V1 by suppressing the activity, right? So it's
Right, but this is what we wanted to do. Phosphine, it could, well, you know, some do masking by just generating phosphines. When you see the flash of light, I don't know, it is an activation, but it could be through inhibition of interneurons, or who knows, you know. And actually in controls, it's not consistent. I couldn't see any phosphines, personally. A bunch of other lab personnel could, but I couldn't see any phosphines, and I was, you know, this was my experiments. I really wanted to see phosphine, so I let them raise the threshold until the barrel distended, and I still couldn't see phosphine. So, yeah, I don't know. This is the nature of science. The interesting part there, the interesting implication of this study was actually for general facial affect processing. And this was actually something that is interesting, and we published this normal data. We are in the process of publishing the negative findings with schizophrenia, but this normal data was actually more interesting here. And what we found here is that in the high spatial frequency condition, the backward suppression was stronger than the forward suppression. Whereas in the normal phases, the broad spatial frequency, the forward was stronger than backward. In the low spatial frequency, there were no significant differences. And let me ask you here again, let you a moment to absorb it. Why would you see, I'll say in an easier way, why would only normal stimuli phases are most suppressed in the fast feed-forward mechanism?
If I may try, because the neural system is optimized for natural stimuli.
Exactly. And remember those face inversion effects, where you see the face in the right position, you process it. That's all those studies that determine that the fusiform gyrus is, you know, especially designed to process facial information and so forth. So this is sort of, I think, this is sort of a similar mechanism that is at work here. When you see a normal face, you process it as a face. And then when you mask it, you kill it right away. But then, when you start messing with it, reducing or increasing spatial frequencies and so forth, you are starting to process it as a regular object. And we're not as good at processing regular objects as we do with faces. So this was actually an interesting finding. All right. So to conclude, social cognition was found to be another important construct in schizophrenia that probably mediates this path from basic cognition to functions. Okay? The last part that I want to talk about is actually dysfunctional cognition. And you might recognize this young man here. This is Aaron Beck. Aaron Beck, and it's, sorry, well, this is me. I was a few columns ahead there then. But this research started from a symposium that Aaron Beck, as you know, is most well known for his studies in depression, right? Beck in triad, and you sort of develop negative beliefs, dysfunctional beliefs about oneself, about others, about the world, and so forth. One person might take an exam and get a bad grade and would think, well, this instructor is an idiot, or I just didn't work hard enough, and he would be okay. Another person would take the test and get a bad grade and say, well, I'm a complete failure, nothing will come out of me. So these are the type of dysfunctional beliefs Beck was talking about. And he was doing it for years in depression. Turns out that in his 70s, he started thinking about schizophrenia in similar terms. and he did some preliminary studies, and he found interesting results. Patients score, schizophrenia patients now, score higher than controls on these types of questionnaires. Schizophrenia patients, just like depressed patients, also have misinterpretations about themselves. And among these patients, higher defeatist beliefs correlated with more severe negative symptoms, but not positive or mood symptoms. Okay? So again, this is not that surprising, but it's kind of cool that it leads to these symptoms that might seem similar to a depression, manifestation of a depression. And they do worse on social and vocational functioning, and they also have worse neurocognitive functioning. So at this symposium, we discussed a lot, and Beck has an amazing amount of energy. He drives you forward. He wants you to try and do these ideas and put into use these ideas that he has. And so we did. And what we found here was that dysfunctional attitudes, we took 109 patients, we did a structural equation modeling technique, and we found that dysfunctional attitude actually significantly predict real-world functioning. And then when you put a negative symptoms in the model, it mediates. Now it's partial mediation. You see, it is significantly reduced but it's not zero, which means that this is important, but the direct path is also important. So now dysfunctional attitudes goes to negative symptoms and ends up at real-world dysfunction. And I promise I will make sense of it because it gets more complicated. Yeah?
Sake of argument, I submit to you the hypothesis that they are truly deeply depressed, and that will explain everything you have there.
Well, but see, it is not associated with positive or mood symptoms.
No, because if you're really depressed, you're not going to move.
You're not going to
move. You're going to be paralyzed. You're going to be bedridden.
Well,
it's real kind of depression where you spend hours and days in bed.
I tend to agree, but this is a nosological question, right? Because in the DSM, you cannot diagnose depression if you have schizophrenia. You can diagnose mood disorder, NOS, but you cannot diagnose depression. But what is DSM? A bunch of psychiatrists deciding what's right and what's wrong. It's fine. This is the best we have. This is the best we have. Psychiatrists are my best friends. This is what we have. Okay. So let me now make the story complicated first and then try to explain it by putting it all together. So now we have a large model, recently been published in the archives, a large model with everything I discussed so far placed in one. Over here we have enough. By the way, one thing about, and you are nicely set up here with all the hospitals around everything to do studies like that. In Israel, there's no way I can do something like that. To collect 200 patients, structural equation modeling requires a lot of subjects. Okay, because each of these has many measures, and when you make the story complex enough, you're starting to have about hundreds of patients. There are other disciplines where they use structural equation modeling, but they just use simulated data. We cannot do that. We need actual subjects. Okay? So we collected about 200 patients, and what we found here in the model, that early visual perception leads to social cognitive deficits. Those are a bunch of masking measures, social cognitive deficits, which are measured by measure of social perception, theory of mind and emotional processing. This is vignettes that they need to extract emotional information. Here they need to interpret the emotions of others and various levels of emotion processing. And this leads to defeatist beliefs about themselves, what they can accomplish, which resulting in negative symptoms and eventually leading to disruption of their daily function. So to summarize the story, the way we think about this. I mean, life is complicated. We need statistical techniques that will allow us to capture the complexity of this dysfunction. It's not easy. It takes a long time. These grants take several years to do. I sometimes envy our basic scientists that can run a study in a few weeks. For us, it's several years. But you need to collect data on a lot of patients and then see if it actually pans out. So the way we think about the story of schizophrenia, how patients end up dysfunctional, why can't they function normatively in the world, is that they first have those deficient perception, cognition, which leads to distress, social and academic failure, and of course, the Beckian dysfunctional attitudes about themselves. I cannot succeed, there is no way, nothing I can do about it, leading to negative symptoms, or the deficit disorder, I cannot do anything, and eventually leading them to poor functional outcome. And of course, in terms of interventions, I've told you the story starting from here. One could, of course, go back and study things like genetics, neurochemistry. All of this happens here. And once we have those technological advances and we could just twitch their genes, everything is solved. You know, of course, we could talk about ethics for several hours, but it's hypothetically possible to intervene there. We don't have that technology right now. So now we have all these mediators, which we can, right? Beck already developed an intervention, fairly effective intervention for depression. And he's doing now with schizophrenia with promising results. We just do CBT with schizophrenics, and they're promising results with schizophrenia, especially for negative symptoms. So each of these mediators, until we have the genetic intervention available, we're still kind of stuck here. Now, and I just want to conclude by a story about the future. And this is a story about neurostimulation. Up until recently, actually, when we discussed cognitive enhancement, the first thing that came to mind is probably some pharmacological intervention, giving a certain drug and it enhances your brain. More recently, there became neurocognitive or psychosocial interventions that apparently improve your cognitive abilities. And more recently, there are some promising studies of neurostimulation. One safe and relatively painless stimulation, even though you kind of administer electric shocks to the brain, is called transcranial direct current stimulation. This one is actually more benign than TMS. TMS is a pulse, is a direct pulse sent to the brain, it's a magnetic pulse, and it causes action potentials. It causes neurons to fire. This one does not cause neurons to fire. It just modulates the neuron. It makes them more readily available for processing information. So this is sort of the theory about this, that it facilitates LTP processes, okay, because it makes the neurons more readily to process information. And that's the grant I got. We're going to stimulate patients' frontal areas, and we're going to see if they longitudinally perform better on various cognitive measures. And next time, not in the near future, I'm sure, but next time I can tell you about that. All right. So just to acknowledge, this is our UCLA lab with Michael Green, Bill Duran, John He, Jonathan, Markley Alcovoni, Alan and Steve Martyr, and several collaborators from Israel and from other US universities that I work with, and I thank you for your attention.